Yes, we are online. People are joining. Okay, we can start. It's 11. Uh, Welcome everyone. This is our 660 international webinar in Bioengine platform. But today's topic is a little bit different. Today we are invited one of the young scientists in Institute of Life Sciences, is at GBT Autonomous Institute in India. He's uh, very good in field. As you know, Soma will introduce at the time with the details, and he's world 2% scientist ranking. His papers and citations has come, and he worked in a very interesting topic, is the circular RNA. And when he did his research, he faced many challenges, and he faced many technicality related issues during his research, and then he tried to solve some of them and to make a company, RNA Bio, and nowadays it's a private company and selling the same products which is he invented. So once again, it's a live example that necessity is the mother of invention. So I request Shoma to introduce Bioengine little bit, then the speaker, Dr. Amaris Panda. Then we go for the short presentations, and our session will mostly focus on the interaction part. We try to know his journey, his companies, his product, how he started, where he gets funds, and all. And I request all the people, don't bother about the certificate. We have a very simple few steps. You have to register for this webinar with the email ID. And today you need to collect the feedback link from the live presentations. This is assure your presence in the live event, and you need to submit this form within 5 hours after the end of the webinar with using the same email ID, because we checked your email ID in registration form and feedback form, and everything done by Google Forms. So it's totally automatic. We are not manipulating or checking anything. We just match. Those matched are found, they get the certificate from our website. So it's simple. It's not a big deal. And yes, it is only applicable for certificate, not for the viewing, because it's free to all and it's available in the YouTube. And after the end of the session, it's still available in same link, same channel. Subscribe to our channel, and let's Soma begin the webinar.
Okay. Hello, everyone. Welcome to another Bioengine webinar. Bioengine is a non-profit organization created to promote plant research worldwide. The webinar series has been designed to build a platform from where plant scientists can present their research to the world. We hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on building the future of plant sciences. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us. We are also thankful to you, the viewers of our Bioengine family, for your interest in our Bioengine webinars, your constant support and appreciation. We have received a huge response through the registrations for today's webinar. You can register for the webinars through our website, bioengine.com, and our social media pages. You can live view all our webinars or watch a recording later on our YouTube channel. Please subscribe so that you don't miss any of our live events. After attending today's talk, you can apply for a certificate of participation via the feedback link, which will be provided after the presentation. Please type in your questions related to today's talk in the YouTube chat box for the Q&A session. The topic for today's webinar is Academic Entrepreneurship Challenges and Rewards by Dr. Amaresh Panda. Dr. Amaresh C. Panda was born and brought up in Jodhpur, Odisha, India. He completed his schooling from Jodhpur, Odisha. He completed his BSc degree from NC College and master's degree from Utkal University, Bhubaneswar. He obtained his PhD in biotechnology from the National Center for Cell Science, University of Pune, India. Dr. Panda worked as a postdoctoral fellow for 5 years in the National Institute of Health, or NIH, Baltimore, USA. He also worked as an assistant scientist at the University of Miami, USA. Dr. Panda also worked as a postdoctoral research associate at the University of Colorado, Denver, USA. He has been working as a scientist at the University of Life Sciences, or ILS, Bhubaneswar since 2017. His research group at ILS is working to understand the role of poorly characterized circular RNAs in muscle regeneration and insulin biosynthesis. The new molecular mechanisms identified during his work contribute valuable knowledge of the post transcriptional processes that influence human health and disease. His research findings have been published in top bioscience journals, including Nucleic Acid Research, Gene and Development, RNA Biology, Cell Aging, Molecular Cell Biology, etc. He also served as reviewer and editor for many prestigious journals. Dr. Panda has published more than 50 articles with more than 4,700 citations and has an H index of 29 in Google Scholar. His research has been highly cited by the research community, making him among the top 2% scientists worldwide, as published by the Stanford University, USA. Besides Dr. Panda's scientific contributions to biomedical research, he has also developed technologies to help research and education in India. Recently, his group at the ILS developed a low-cost RNA isolation kit with superior features to help researchers and academicians. He transferred the technology and became a non-executive director of a startup named RNA Biotech Private Limited, located at the DBT ILS Bio Incubator. Dr. Panda's company is committed to providing high quality and affordable molecular biology kits that accelerate biomedical research even in colleges and universities with low income and technological setups. We are highly honored and very happy that he has accepted our invitation, and we hope all our viewers are there and are ready to listen to him talk. Dr. Panda, we are ready, and the Bioengine platform is all yours now.
Thank you, Soma and Shobrat, for this invitation. And I'm really glad you invited me to this platform where you can find people all over the world, and you are doing a great job in promoting plant research worldwide. So thank you again, and thank you of all the viewers who have joined and who will be joining to the talk. Now, So basically, this title of the talk is academic entrepreneurship. But instead of starting with entrepreneurship, I will start with my academic journey. And as a scientist, my priority is the research. And let me share my screen.
Yes,
Is
it's
it visible?
visible. Yeah.
Okay. Okay. So yeah, as a scientist working at ILS, so my priority is research in the lab, and this research eventually led to development of a product as Suma already described. So I will take you through what my lab does exactly, and some basic things on RNA biology. My lab mostly works on RNA circles. So before going to RNA circles, I will take you through what is RNA and how they are made, what are different types of RNA, and then we'll go to how we come up with a problem and we try to solve the thing by developing a kit. To start with, like as you all might be knowing, like DNA, RNA, and protein are the life molecules. So basically, always it is believed like DNA is the life molecule, and it is packaged in the chromosomes. Like we have 23 pairs of chromosomes, and if you really look into the size of cell, chromosome, and the DNA, you will be surprised how long our DNA is. So basically the cell is only few micrometer long. And inside the cell, you have nucleus, and inside that nucleus, we have a tiny chromosome. And inside that, we have about 2 meter long DNA coiled in it. And you can see this coiled DNA is in the chromosome is coiled around the histones, and if you stretch them out, you will find all ATGC. And the length of human genome is about 3.2 billion bases, and it is about 2 meters, as I mentioned. And you will be surprised to know that, like, if you remove all the DNA from a human being and stretch it, it will be more than 10 billion miles. And that to keep bring it to perspective, like you can go and come back from Pluto 17 times. So you can imagine how big our DNA is in a tiny nucleus, how much DNA we are having. And then what is the function of this DNA? I said this is the life molecule. Okay. So if it is the life molecule, then everything should start from DNA. Yes, that is true. So the DNA is the life molecule, and that gives rise to RNA and then protein, as I showed in the first slide. So this double-stranded DNA in the nucleus is transcribed to give RNA. And most of us are very aware that DNA makes a RNA that is called messenger RNA, because that takes the message from DNA to make a protein. And that process is called translation. So the messenger RNA brings the message from DNA, and that is getting translated into a protein, and that is the functional unit. Most of us believe this is what is happening. But if you really look at the genome, you will be really surprised that the things that is getting into the translation, or the part of the mature mRNA that is giving rise to protein, is hardly 2 to 3% of the genome. But we always believe it is DNA to RNA to protein. But then what does the other part of the genome is doing? And in the last decade, what happened is the Human Genome Project. When the Human Genome Project was complete, the next generation sequencing came in, where various bioinformatic pipelines developed to identify all the RNA that is coming from the genome. And people were really surprised to see that more than 85% of the genome is actually getting transcribed, which people are thinking, okay, few percentage of genome may be functional and the other part are junk. And some people even term that junk DNA. So they are not functional. But with the NGS and high throughput analysis, people found that more than 85% of the genome is transcribed, and most of them are not coding for the protein. So the conventional belief of like DNA to RNA to protein was challenged. And this non-coding RNAs, it is not that last decade only identified non-coding RNAs, they were discovered decades ago, but the number and the type of non-coding RNAs discovered after the next generation sequencing was huge. And that led to discovery of hundreds or thousands of different novel RNAs of different types like microRNA, piRNA, snRNA, snRNA, tRNA, H1RNA, and there are like yRNA. And recently there was something called circular RNA was also discovered, and long non-coding RNA, antisense RNA. So the number went on and on. And if you really look at the transcriptome and ask the question, how much is actually functional? So you have tons of RNA, and if you look at the different types of RNA by mass, what we will realize is like most of the mass of RNA is coming from rRNA, that is ribosomal RNA. And the next abundant one is the transfer RNA, tRNA, and the next one is the mRNA. And then the other RNA contribute to a very small fraction of the total RNA pool in the cell. But if you talk about the number, how many number of each RNA type expressed in a cell, then tRNA comes to the top. So in any cell, more than 80% RNA by number is actually tRNAs, then followed by the ribosomal RNAs, and then the very small percentage comes for mRNA, snRNA, microRNA, and other link RNAs. And what this tells us is that, like, although mRNA is, we believe that is giving a functional protein, the system makes a lot more of non-coding RNAs, and it cannot be junk. The evolution cannot be that foolish to make all these non-coding RNAs and they don't have function. As you can see, the tRNA and rRNA are integral part of the translation system. Without this tRNA or rRNA or any modification or any mutation or problem with the formation of this tRNA or rRNA can lead to dysfunctional protein synthesis. Or the mRNA will be there, but you will not have the functional protein. So they are critical for making the protein from mRNA. So basically, although the mRNA portion is low, and that gives the functional protein, the other non-coding RNA, which forms the majority pool of the transcriptome, are actually regulating the formation of the protein. And it is not only involved in the formation of protein, they can also do other functions. Not only like for the translation, they can also serve as the genomic information. Like RNA was the first genetic material during the evolution. And now if you see COVID virus, the genome is RNA. HIV, the genome is RNA. So still we have viruses which have genomic material as RNA. And then for the structure, ribosomal RNA. Without the ribosomal RNA, ribosome cannot have a structure, because the structure comes from the secondary and tertiary bonding of rRNA that forms the skeleton of ribosome, and then the ribosomal proteins assemble to form the ribosome, what it looks like. And then the catalytic property, like RNA, like group 1 introns, or ribosome itself, that has enzymatic activity. So that is catalytic activity for the RNA. And then the regulatory thing. As I said, most of the RNA in the cell, they are not coding for protein, but they are somehow involved in regulating the protein formation or structure or catalysis, and eventually the expression of genes that leads to development. If we categorize them, like mRNA is on the top, that gives rise to the proteins, but rRNA, tRNA, they are integral part of the translation system. And then we have hnRNAs, scRNAs, snRNA. These are also all are regulatory RNAs. They either involved in RNA processing or splicing, and like snRNA, SNO RNA, they are involved in RNA processing and splicing. And the regulatory RNAs like link RNAs, microRNAs, or the recently discovered circular RNAs, that is circRNA, they are involved in regulating transcription, translation, and stability of the mRNA, which is actually giving the protein. So the bottom line is, although the RNAs are non-coding, majority RNA in the cell are non-coding, but they have functions. Without them, the protein cannot be made from mRNA. And they are involved in general translation of proteins, or in a regulatory way, they regulate the protein synthesis. And as I mentioned, like the newly discovered circular RNA, although I said newly discovered, but it is actually not new. It was discovered like 4 decades ago in electron microscope. And people saw that there are RNA which look circular. And the first thing people thought is like it could be intronic lariats, which are known to be RNA circle, but they are eventually degraded without having any specific function except for the splicing. Then with time, there were few discoveries. They saw RNA as circles, and they saw RNA exons are arranged in a different way, and some people even named it as scrambled exons. So that like in a RNA, you have exon 1, 2, 3, 4 to make a mature RNA, but they are scrambled. Exon 2 is coming after exon 3. So people thought these are maybe linear RNAs, but they have misarranged scrambled exons. But as I mentioned, like this in the last decade, when the NGS came in and there were novel pipelines, bioinformatic pipelines, they analyzed all kind of RNA, all kind of possible arrangements in RNA fusions in different types of RNA. Then people discovered that there are many RNA which show the pattern like scrambled exon. And then people with different biochemical methods confirmed that these are not actually scrambled exon, these are actually a circular RNAs. So the RNA doesn't have end. So the It is a head-to-tail joining of RNA fragments that makes them circular. And after that, there are hundreds of articles in last 10 years to establish that these RNAs are ubiquitously expressed and they're functional in various systems. And as I said, like these RNA circles are generated from fragments of RNA. As you see here, if the primary transcript is here and it is transcribed from the DNA, it have two ways to go. Either it goes for canonical splicing, where all the exons, one by one, they join together to form a linear mRNA. That is the conventional RNA we always talk about. Or the RNA decide to go in a different way with head-to-tail joining. Either this exon joins to the start of its own exon, or it can join to the start of the exon upstream to it. And similarly, many combinations are possible. And based on these combinations, people discovered that circular RNAs can be coming from exonic regions, and they call them exonic circular RNAs. They can come from exonic circular RNA with the retained intron. So that those are exon intron circular RNAs. And then the other category is the splicing lariates. And sometimes the lariate are very stable, not degraded by the deep branching enzyme and give rise to stable RNA circles. They are either called circular intronic RNAs or intronic circ RNAs or cis RNAs, stable intronic RNAs. So these RNAs basically form from part of the primary transcript, either from exon or intron. And how to detect them? If you have RNA coming as circles, then what is the way we should detect? For linear RNA, we always put primers to do PCR and look whether they are aligning with the genome in a linear fashion. And we call them, okay, these are the linear RNA. But for the circular RNA, what happens is there is no end. So to characterize them, like what people have come up with is divergent primers. Divergent, as I said, like you have a RNA where the exon 2 comes after exon 3. So if you put a primer between exon 2 and exon 3, and they're facing outwards on a linear RNA, so they will never make a product if the RNA is linear. But if the RNA is a circle and these two exons join together, you can have a PCR product. So that is one way to detect, and it is one of most used method to detect or quantify circular RNAs. However, there are other ways like northern blotting, 2D gel electrophoresis, gel trap, and exonuclease treatment. Like exonuclease is basically used to degrade the linear RNAs where the circular RNAs are left behind and they are intact. And these RNAs, enriched RNA, can be taken for sequencing or for PCR for validating a RNA is circular or not. So as I mentioned, like this RNA can be taken for RNA sequencing. So we need to understand how the sequencing works actually. So in the sequencing, as I said, the genome gives rise to a pre-mRNA that goes for splicing to form mature RNA. And these mature RNA are fragmented into different fragments because the sequencing machine cannot read more than like 150 or 300 bases. Although there are methods where you can do long read sequencing, but the most used method is the Illumina sequencing, where the RNA are fragmented and then you put make a cDNA, put the adapters, and then amplify them. So if this is the actual sequence of the RNA, so after fragmentation, you will get different sequences. And then after getting the amplification and run it on the sequencing machine, you take the sequence from the sequencer and then align with the genome and see where they are aligning. And based on that alignment, so here you can see only 3 exons. So that means this mature RNA have RNA fractions, RNA in these regions, and the middle region have less number of reads. What that mean is there are RNA where you have 1, 2, 3 exons. There are RNA from the same region of the genome that has 2 exons. The middle one is missing. So that is basically called a splice variant. And so during splicing, 1 exon is not included. So this is how the linear RNA are identified from RNA-seq. Then how to identify circular RNAs? So there are different pipelines. So this is one of the pipeline we mostly use for identifying circular RNAs in our lab. Like this pipeline was developed few years back by another group. And what they did is they used the RNA-seq reads and split the read. Instead of matching like here, you are matching the read to the linear genome. What they did is the reads were split into 2 fragments and then matched with the genome and looked at whether the end of the read is coming upstream to the start of the read. And that way, they could figure out these reads are half of the read is coming at one end and the other half is coming at the other end. So that suggests that probably these 2 ends are joined. Because of that, you are getting reads which are a combination of this exon and this exon. So in that way, they identify the circular RNAs and annotate them and saying, okay, this circular RNA is coming from 3 exons, this one is coming from 2 exons, or this is coming from 1 exon. Whether it is from the exon or intron, that is also annotated based on the read matching to the genome. I'm not sure if everyone got it. So to generalize the concept how RNA-seq we identify circular RNAs. So let's see this sentence. Like we all know Bioingene is promoting plant research worldwide. So if I want to do RNA-seq for this RNA, I imagine this is one RNA. And after fragmentation, you can find many fragments promoting plant research worldwide, Bioengine promoting or worldwide Bioingenes. So these are different combinations after the fragmentation you get. And when you align with the initial sentence, because this is our reference sentence, we have the reference genome and we know Bioingene does this, and this is Bioingene promoting plant research worldwide. So we align the reads to the known sentence. Sorry. So, so after aligning, what we get is this, the same thing. And the surprising thing here is there is one word here, worldwide Bioingene. And if you put that to the sentence we have, it is not matching actually. So either it can match to the beginning of the sentence or to the end of the sentence, but it is not fully matching. So That's where the split read comes into picture, as I described in the last slide. So what it means is, probably this sentence, what we are reading is linear. Probably this might be reading as worldwide bioengine promoting plant research, worldwide bioengine, if we put it together. But that is not the case. We know this is the genome, this is the sequence, and this is the order we should see. If it is happening this way, then what is the possibility? So the possibility is this. So Bioengine promoting plant research worldwide. So this is worldwide and this is circular. So anyone can read from any word they want. So there is no end. So this is what my lab works on. On RNA, they don't have any end. And till date, there are huge number of circular RNAs have been identified. And these are mostly from human samples, although there are many circular RNAs identified in mouse, fly, monkey, and rat. So these are basically the databases which actually enlist the circular RNAs identified by RNA sequencing. And as you can see, the number is more than 1 million. So that is far more than the number of genes we have. And this is happening because one gene can give rise to multiple circular RNAs based on the batch splicing or head-to-tail splicing it is going through to give different combinations of circular RNA. As they don't have any end, every RNA coming from a different region from any mRNA are different and they could have different functions. As I mentioned, you have hundreds or thousands of circular RNAs, and as of now, it is very well established that these are abundant, ubiquitously expressed, and there are also many of them are conserved across various species, and some of them they show tissue-specific expression as well. Just because these are coming from mRNAs, so as tissues have specific mRNA expression, they are also coming from the same mRNA, so they have tissue-specific expression. And yeah, excuse me. Yeah, so what is their function? Like if we have more than you say 1 million circular RNAs are known in human tissues, what they do? And in last 10 years or 11 years, people have found that circular RNAs, they are very stable, so they can bind to microRNAs and act as a sponge for microRNA they are targeting. They can also bind to RNA binding proteins to regulate translation, or they can also regulate splicing. And there are also examples where circular RNAs bind to transcription factors and regulate transcription. And recently, many articles have highlighted that since these are stable circular RNAs, they can also act as biomarkers for disease detection. And they are also secreted in the exosomes. Sorry about that. So, and recently, although these circular RNAs are known to be non-coding in nature, people have found that many circular RNAs, they have internal ribosome entry sites that can have translation potential. Even some are modified with M6A that helps in translating into protein. And as you can imagine, if the translation starts, and it will be very efficient for a circular RNA because it can just go round and round. So these are the functions so far known. And my lab at ILS is mostly focused on how these circular RNAs are regulating muscle regeneration or pancreatic beta cell function. So one of the major grant in our lab that focuses on how circular RNAs can regulate beta cell function. Pancreatic beta cells are the key for insulin production from pancreas in response to glucose, but with different conditions, and for long-term high glucose lipid stress or genetic issues, these beta cells fail to produce the insulin the body needs for glucose homeostasis. And in a long run, the insulin production goes down and the glucose level goes up. And basically, that is what we call it diabetic. And as you know, it is one of the major diseases all over the world. And we want to understand if these high glucose or lipid conditions affect beta cell growth or apoptosis that leads to development of diabetes. So my lab focus is to find circular RNAs. They have role in development of diabetes, specifically looking at their function in the pancreatic beta cells, which produce insulin. To characterize these circular RNAs, so we need to isolate them first, right? As I said, there are many methods to identify them, but there are not a single kit in the market to isolate them. So what we thought of is like from my postdoc days, we tried to find a method that can isolate circular RNA. So what we developed is it's called RPAD. So where you treat the RNA with RNHR so that the linear RNA is degraded. Then you add a poly-A tail to the RNA which are not degraded and have ends. And then deplete them so that all the linear RNA having poly-A tail or not, they are either degraded or depleted. So leaving a pool of enriched circular RNA. And this can go for either RNA sequencing or RT-PCR validation to see what are the RNAs expressed in a cell. But sorry. So, but this process is very tedious and it needs enzymes for degradation, then purification. So these steps add a lot of time and money for this process. So eventually we thought like we should have a method for isolating circular RNAs. But when we are trying to develop, like as I mentioned, one of the method to see a circular RNA is through gel trap in one of my slides. So we were trying to develop that method to have a easy and cost-effective solution for circular RNA isolation. But during that process, we realized that after any method, whether it is enzymatic or the gel trap, you need to isolate the enriched circRNA pool from the matrix. And what the problem is the column-based thing. Because after you do all the treatment, you want to isolate. So that is not only the problem for circular RNA isolation, it is a general problem with any RNA isolation anyone does. So most of the kits available in the market nowadays are column-based. So those are basically silica column based kits. So, and they're not scalable. What it means is like you have bought a kit for 50 reaction. So they have given you 50 columns. So you are limited to do 50 reactions only. You cannot do 10, 100 reaction, or you cannot increase the capacity. If you have sample of higher size, you want more yield, you cannot do that. So you are limited to one column and then mentioned number of cells you need to use to get a specific amount of RNA. And then since this is column based, you need to elute the RNA in higher volume, because it has to take like 30-40 microliters of elution buffer to make the matrix wet so that you can get your RNA. And since it is column based, it also needs centrifuge, and that adds a lot of cost and electricity to the lab. And sometimes like centrifuge is occupied, or it is and sometimes it's not working. So those are problems. And then the other problem with any RNA, whether it is a circular RNA or total RNA isolation, the genomic DNA contamination. That always comes with the RNA. Some kits, they provide DNAs for on-column digestion, or some say, okay, after the isolation, it can be treated with DNAs. That adds cost. And most of the column-based kits are for total RNA. When they say total RNA, that means RNAs mostly above 150 or 200 bases. And these are mostly long RNAs. And if you need to isolate small RNAs like microRNAs, then you need a special kit for microRNA isolation. So looking at all these problems, although we could not figure out how to make a circRNA isolation kit successfully, but we come up with all these problems which were there. We never thought of having it because we are used to it using the column-based kits. But when we are trying to develop another kit and easy method, then we realized these are real problems for any RNA isolation. And then we thought, like, what is the solution? Then what we did is, like, we thought probably we should make a kit that is that can isolate DNA-free total RNA, including microRNA, so that people don't have to buy multiple kits for long RNA or microRNA, and other issues also need to be sorted. So what we did, we developed a method like where we isolate the RNA from the cells, and then we use magnetic beads for RNA isolation. We remove the genomic DNA by centrifugation, and then whether it is long RNA or total RNA or microRNA, those can be isolated with magnetic beads. And as you can see here, so if you isolate total RNA, you can also see the small RNA fractions, microRNA, tRNA, and there's 5' rRNA. But as I mentioned, most kits use the column-based method, which is mostly for the long RNAs. So you basically lose those small RNA fractions. And after developing this, the idea came like, what to do with this method? Like, we can either publish it or make a kit, or then we realized, like, okay, first thing as a scientist we did is we published, and then we thought, like, okay, let's make a kit out of it. And then we transferred the technology to the company RNA Biotech Private Limited, and last year, actually, after we gave this kit to few people who wanted to use the prototype, and we got the feedback. And based on the feedback, we developed the kit into a packageable form and a viable product. And we launched it in last year with our late ILS director, Dr. Ajay Parida, and the Minister of Science and Technology of Government of Odisha and Startup Odisha people, and our bioincubator team, Dr. Nivedita. So we launched this product and it is already in the market now. And the key features of this product, as I mentioned, these are the problems with the column-based kits in few slides back. So we used magnetic bead-based isolation, and since it is a magnetic bead-based isolation, the RNA can be eluted even in 10 microliter. So, and then the genomic DNA is not a problem. You don't need to treat it with DNAs, and it is also capable of isolating microRNAs and total RNA based on the need. And it is easily scalable because it is magnetic bead based. So based on the requirement, based on the amount of sample someone wants to use and the amount of RNA he or she need to isolate, that can be easily scaled based on how much buffer or reagent you need to use or how much bead you need to use. The 50 reaction kit can be used for 100 reactions, or it can be used for 10 reactions also if you need more RNA from one sample. And finally, this is my team. This is my RNA biology lab at ILS. And I really thank my student who really helped me in establishing the lab and developing the product and taking that technology to have a startup eventually. And then the funding agencies, intramural funding from ILS and extramural funding from DBT India. And the major funding comes from Wellcome Trust DBT Alliance for the lab, and that is going on. And I thank the lab members as well as the funding agencies for the research. And thank you all for your time, and thank you Bioengine for this opportunity to share my science and entrepreneurship journey briefly, and thank you.
Thank you. Thanks to you also. Actually, it's our pleasure to meet someone who did research and try to convert, and not only the publication, also make a product from that research, which will be very helpful for countries like us and other developing countries, because it's a low cost and effective. So we are always searching for such product, but those not sometimes those are not available in our country, or maybe it's a high price. So it's a very good approach. It's not benefit science; it's also may benefit society. So congratulations to you and your team for developing
Thank you.
this, and I'm sure many more product will come from your end. As you as I'm sure everyone would agree with me that people work on RNA is little bit lower, and few people are working on RNA due to stability and isolations and everything,
It's
contaminations.
difficult to work
Yes,
with.
and their lab also very need to be very uh, contamination free or dust free. So yes, it's a challenge to work with the RNA, and specifically when you work in a special type of RNA. Today's those are graduates, undergraduates, and PhDs are will be very benefited from your talk, and as I can see from the YouTube live chat, they are enjoying it, because in a one presentation we can see many more type of RNAs are there. So obviously we are not aware as much aware uh, on the others part of molecular biology. RNA is very interesting. Always it's shows something very important, coding or non-coding. Both are RNAs are nowadays get the importance. So let's we start your entrepreneur journey a little bit more. And I welcome all the participants to put your questions in YouTube so we can ask in a live discussion. And also, if you have any others related questions to today's talk, we will put into discussion. But yeah, sure, we can't choose each and everybody's questions, so selectively we choose the questions. So I have few questions already for you. So we are going to go one by one, and this is a little bit discussion type, and not need to be so formal in that
Okay.
way.
Okay.
So people are relate to you, and those are neighboring countries participants. As I can see in YouTube chat, lots of people from other countries. But here we try to focus on the questions which is there is no barrier of a countries, but obviously we put examples from the India, where the we can how type what type of funds are available and all. And I believe all the country have this type of funds and others through the other different name of agencies. But yes, every country has a aim to promote entrepreneurship, to promote science, to develop science. So yes, it has to be there in a different name. But for an example purpose, you can use India's agencies and funding informations. But others few other questions like the how to write a grant, how to publish a paper, how to select a manpower. These are the
Yeah.
common. These are
Those
the
are
yes. These are the
irrespective of the
yeah,
country.
any
Yeah,
countries. And
because I have no idea how the funding and other things work in
in a
other
different
countries.
yes, it's a
So
very different difficult to say
right,
any one which is not
right,
from that country, or he has to be some international policy maker. So we are not
right,
today's we
right,
are not in that discussion. Our discussion is mainly for the your journey, your challenges, and your achievements. And we going to learn it many things from this discussion, I hope so. So let's start the interactive session. And in the
okay,
meantime, people are putting their questions in into live. So in the end, I will take some fewer questions from that if we miss in our discussion. So uh, my question is but it's not a question, because we are going to give it a different section. It's a different section than your presentations, because in future we are going to plan to cut the videos and promote the
right,
discussion these
right.
sections differently. So maybe some you need to be some repetition from your presentation also. So just I want to know how you already mentioned, but still wanted to know your how you build your career. Means I'm understand that you are from a village in Orissa, and now going to the PhD from a reputed institute in India, do the postdoc from the foreign, and now you're a scientist in a central government DBT institute, Department of Biotechnology, Government of India institute, and it's one of the leading institute in Orissa state. So it must be not a simple journey, or it's not must be a easy journey. But I want to know, and it give our some motivation to all the speakers, listeners, or participants, that what how you build your career. and what is your interest to do in the science and come to the science in that way.
Yeah, this is very interesting. Uh, so if I go back to my school days, as I said, I'm from a village. Yeah, so I come from a very small village in Jajpur, and not probably, I'm the only one who could get a PhD and who could go out of the country for job or training from my village or even nearby villages. I think I'm the only one. And the journey is not that, I would not say it is difficult, but it's a journey. So I won't say it is challenging or difficult. And uh, from the schooling, like if I start, like my school didn't even have a bench to sit and even didn't have electricity. No, not during my time, even years after I graduated also many students after me also went through the same thing. And even we didn't have a road to go to the school till high school, like you go through the farmland. And you can imagine in rainy season you can have holidays if you fall somewhere. And so this is not difficult, this is also fun, this is experience. But the only thing is how focused you are onto your target. Like if you are a student, mostly I would say for the students, it doesn't matter where you come from, what is your background, what school you are reading in. Most of the time we are very fascinated about, okay, we want to go to the top school, we want to go to a school having all the facilities. And eh, 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 although these are important things for students to have facilities, but even if it is not there in some school, you should not be discouraged. Like you should think that the facilities in the school doesn't make a good student or doesn't make a successful person at the end of the day. It is like, it is who you are. And if you are determined to do something, it will happen. And school, any school, even in the top school, students also fail. So should we blame the school or the student or the teacher? You cannot blame any one of them. It is like some part everyone is responsible, but the for the success, I think it is the student. And even after my schooling, when I went to college, like school was completely in Odia, and you landed up in a college where the textbooks, everything is in English. And then you realize that the things you read in your school are no more useful. The terms you know for a cell or nucleus or DNA, they are no more there in that book you are in the college. And then then another struggle start to learn everything in English. And after college, you go through the exams and to find a good place in master's degree. And when you come to the master's, you then, like you said, I'm from a village. So when you are from a village, you have that village instinct with you. And we always feel like, okay, we kind of inferior to people who are coming from cities because they speak good English, they are smart, they are very vocal. And people from villages, we are very like kind of like we are not coping up with the situation. I think not me, many of the listeners must have gone through that. Like you feel demoralized. But at the end of the day, when you go through national level exams or even your university exam. It doesn't see your smartness or how good you are speaking in English, or it only look at your knowledge. If you have done your job correctly, you have read the books thoroughly, so that matters. And then when I moved to PhD, like I went from Utkal University, uh, as it is a basic science department from zoology, we didn't have much of the equipments, the modern equipments, and we are not exposed to even, you say, pipetting. We didn't run a single check, and you land up in a high profile institute in India just because of your knowledge. But you don't have any exposure to any equipment. You have never seen a PCR machine. You have never handled a pipette to run a jet. And people are from different background. And if I go from Odisha, I know how people are in Odisha. They talk in Odia, and they all are known kind of people. But when you go to a national institute, you realize you have people from all over the state with a different background, different language, and that diversity. So accept the diversity and learn. It is also a challenge. And specifically when you don't know handling equipments, or you have not done yourself any experiment in the thing. But slowly, with patience, even some people even said, like, probably you will not survive in this competitive things. You may not survive the PhD journey. As many PhD students realize, like, it's a very difficult thing to do. But if you are determined to do, things will happen in your way. And you have to be just patient and hang in there. There is no other way. And eventually I published papers, learned pipetting, learned how to do experiment, then went for a postdoc. And then when I landed in NIH USA, that culture is completely different. Now we had people from different states. Now we have people from different countries itself. You have 10 people from 10 different countries. Their mindset, their work culture is completely different. And the US work culture is also completely different from Indian scenario. And also the language. Although we speak English, but our English is different than what is there in US. So the first thing you realize is that people don't understand what we are saying. And when you have different people from different countries, their language is also in their way. So you also struggle to find, cope up with that change in environment and this thing. And then eventually, after a few years in US, I thought, like, okay, now let's go back to the home country and do something. And as our government is always focusing on, okay, make in India and Atmanirbhar Bharat. And so I thought of, like, okay, let me go back and do what I need to do. And if I stay in US as a postdoc or a research associate, I will be doing what the mentor is asking me to do, because it is his project, his money. I cannot do what I really want to do. And if I have some ideas, and that is my idea, then I should have my own grant to do the research and to have my own lab. Then I look for the opportunities, how to come back to India. And then I applied many places, and fortunately got here and came here. And then I got some good grants and established a lab, and eventually Then, as you already mentioned during introduction, the necessity is the mother of invention. We realized problems in our lab. We are not focused on solving um, others' problems, although it will solve others' problem. But the first thing we started with what problem we have and how we can solve that, and we did that. And yeah,
Yeah. And
thank you.
thanks for your this sharing this journey. Actually, why I'm putting these questions, because I'm also from a village, and many people in the village or in small town, they are thinking the same thing, that it's not for me. I can't do that. I can. It's not possible for me. But at the end, it's matter of your intention, your passion, and your
Your
dedication.
perseverance, basically.
Yes,
Yeah.
perseverance is matter. It doesn't matter from where you are, because nowadays many papers has come those countries where the English is not get the importance, because there a lot of things are changing right now. You can uh, write a paper in your own language and send it to a company for the money, and they can convert it. It is actually in English.
Suitable language
Yeah,
and
suitable
make it.
language,
Yeah.
and not only the grammatically correct, is also academically correct.
Yeah,
So
correct.
it's a nowadays it's possible. So just your intentions is matter. And as you said, there's some problem which is not a problem for a developed country or Western countries, like availability of uh, uh, electricity, or availability of number of basic instrument like centrifuges or anything. So
Yeah.
sometimes it's problematic for like developing or these Asian and South Asian countries, because
Yeah.
we share a same instrument for many peoples. So sometimes it is a problem, but which is not look like a problem for others countries or anybody wants. So it's a very specific country to country, very specific to lab and infrastructure to infrastructure. But yes, your journey um, definitely motivate many of us. And thanks to share
Thank you.
from the beginning to NIH, and different times, different times, you have got different challenges, and I'm sure you overcome it. And thanks to come back in India, because it's also
Yeah.
every countries is a dream that people goes to the others countries, learn learn some techniques, and come back to their own country and use these techniques to build their country's economy. So you are doing growth in that. I'm sure.
Thank you.
So next, my question is: it's proven that your interest bring you here, and also as you already listed in top 20% in scientist, based on your publications. So it is understandable that you not only do the science, you also uh, did some good publications. So my question: can you tell us about this? How you done these things to, and how it is noticed by Oxford, for the Stanford for the 2% scientist? So how your name came into the 2% scientist?
So, yeah, so as a scientist, what we do is whatever discovery we make, like we put it in the form of paper, uh, articles basically. And when we put the articles online, many people read it, and some people use them or cite it. So that is what called citation, basically. Like how many people used the research what I have published. So if 10 people are uh, using my paper and cited in their publication, so my paper gets 10 citations. So based on that, and in each field, the citation index also differs. because plant science may not have that many citation as compared to cancer biology.
Yes.
Or if we compare to physics, it will be different. For chemistry, the index will be different. So based on the subject-wise distribution, the one group in Stanford, what they did, they took the Web of Science data for each year. For last 3 years, they have been doing that. They take all the data listed in the publications listed in Web of Science and take their citations and the area they are working on. And they look at whether this, suppose my publication has 10 citations, whether it has one author or 10 authors or 100 authors. So based on that, they formulate a weightage scaling system to evaluate what is the importance of this paper in the field based on the number of authors and number of citation and the time scale. If a paper gets 10 citation in 10 years and one paper gets 10 citation in 1 year, so obviously the paper having 10 citation in 1 year is more important. So based on that criteria, having many indexes, they formulate a scale and they selected who are the top 2% scientists in each field. And that listed me in the top 2% in the developmental biology field in biomedical science group.
Okay. So it is means the it has to be very cited publications. It need to be very cited publications, and also you need some publications which can be beneficial for others, like techniques based. So maybe
right.
it is one reason that you got so many citation
So,
because it's technique based.
like it is like more citations in a shorter period of time.
Yes.
So
And
that basically makes your index better. It is not only citations. If the only citation is taken, then my citation will be nowhere. It's
I understand. And you, as you right correctly says, that it's also depend on the topic to topic. In plant
topic.
science,
Yeah, correct.
in plant science also it's depend on the crop to crop. Like in plants, rice, wheat, maize has the many citations, but where the other crop has the lower citations because
Right.
people are working in lower numbers on
Yeah.
that crops. So okay, thanks to for sharing these things. Now we come to the your. I wanted to just know about your dilemma because you did the commercial research also and also the basic science research. So and you got the funding for both commercial research and the basic science research. So which one is more important to you? Means you did both the research, then which one is more important to you?
Yeah. So I think both are important. So if you look at in a way, like these are two sides of a coin, actually. And nowadays, many people say translation, and more funding is eventually like there was one article in Nature recently came that highlights the shift of basic research funding to applied research or translational research, because people realize translation research has significance for the society in immediate future. Like we say, vaccine for COVID. Okay. So that translational research, like we have vaccine within a year of COVID came, right? And that is not because of translational research. That is because of 50 years of basic science research that build the knowledge based on which a vaccine could be made within a year. So basic science is basically the base for any translational research or applied research. And In ideal scenario, if you ask me, it should be a balanced one, and probably more funding should be given to basic research, because basic research not only repeat, many times we think, okay, this is a repetition of the previously published work, so there is nothing new. Of course, there is nothing new in that, but it validates the previous publication, which is very important for the next person to believe whatever is done is correct, even if it is not adding anything new to the society, but it is validating the existing knowledge. And that is very important for anything going for clinical or trial, because if one publication giving a new idea that cannot go to trial or to humans, that needs solid background and solid basic fundamental research to prove that it works in certain way. And if you imagine anything, electricity, magnetic field, anything you name, even the CRISPR technology, one never wanted to discover them. They came out of basic research. It is only that you have to keep your mind open to basic research, thinking that, okay, this is basic research. I'm trying to find the answer for a basic problem in a biological system, and this can be translated. So these are not very different things. You need the knowledge from basic research to translate into a product. Without basic research, you cannot have a product, or if you cannot have a product out of basic research is also waste. So you have to have both things. You have to have basic research that can be translated. If you think it cannot be translated in any way, then it is not useful. But if it is generating knowledge or validating the previous knowledge, that is good enough. And that is why most advanced countries, they are not advanced because of translational research overnight. It is because of their investment in basic research for years. And that gives the knowledge base and the patents that come out of this basic research, that gave rise to industries, products, and other things. So both are important, but people should not think basic research has no value. No, it may not have value for today, tomorrow, day after tomorrow, but certainly it is the basis for any translational research we are doing.
Yes, yeah, correct. I also agree with your thoughts, because, and that is the reason we call it research.
Research,
So
correct.
it's obviously we can start, as you told, that CRISPR, it's got the Nobel just 2 years back, but that thing is starting very long back. So
They never wanted to discover
yes,
CRISPR
that
and
thing
get
is,
a Nobel Prize.
yes, but it's got the Nobel Prize. And I believe it will same happen with how we are advanced in our NGS system. If better system has come into the market, then we also need to re-sequenced of human genome, re-sequenced of rice genome, because then the platform is different. So maybe it adds some more information on that. But if we say that no, human genome is done and the rice genome is done, it's not the right way to say in that way.
Yeah,
So yes,
correctly.
maybe
Like human genome was done like 10, 12 years back, right?
yes.
But 20% of the gap in the genome was filled just last year, right? So nothing is complete. Like I said, circular RNA. When you do RNA sequencing, you discard 10% of the reads, thinking that those are junk, just because you don't understand what they are.
Exactly.
When the pipeline came and they found, oh no, these are not junk. They have something. there.
Mm.
So it is not that basic science will like give you product, but it will help us better understand the system. And the biological research is not that old. We are far away from understanding the system. If we would have understood everything, we would have cured cancer, diabetes, and everything. We are nowhere close to curing any of the major diseases of the world. So it needs in-depth, more basic fundamental research to understand the whole biology.
Next we come to the next questions. It's the, as you got many funds, so I'm sure you just write a fund and got the fund. It's not happened to anybody's life. So lots of feedback, lots of comment, lots of suggestions you have during your times during, and how it is, it encourages you for doing more science, or how you use this feedback as a positive way in your life?
Yeah. So to start a lab or doing anything, you need funding. So have to apply. And in India, I would say like there are many funding agencies like Department of Biotechnology, Department of Science and Technology, SOR, and Wellcome Trust DBT India Alliance. And there are many more grant agencies. So for biology, you can put your grant for any of these grant agencies, and then they look at what is their mandate and whether the grant is fitting to their mandate and the PI has enough expertise to execute it. And like one example, I can say, suppose my Wellcome Trust India Alliance grant. So that grant takes a year time, but the beauty of this grant is like you get lump sum money to do the research the way you want. And like many grants, like you said, basic research and applied research. So I applied the grant in the basic research. And basic research, other name is fundamental research. And when we put a hypothesis, we don't know what we are going to discover, or we are going to discover nothing actually at the end of the day. And the grant agency should have that motivation to take challenging projects. And mine was also kind of that. Even in the interview, people said, what if you don't succeed in whatever you are proposing? You are saying, okay, RNA interaction, but you don't find any such functional interaction is happening. But I feel that is part of the game. Like, and it is, you may discover nothing, but there is a chance you may discover something new. And that is the beauty of basic research. Like some information you discover that no one knows in the world. Like only you have that information. That is the joy of science. And a grant also look at the deliverables. Whatever you have proposed is you are doing it or not. Like every year you have to report them based on your timelines, whether you have done that. And with this Welcome DBT, one thing I would mention is like the grant is flexible. So you propose 3 crore for something, and by the time you get the grant, you already got that machine from some other sources. You need that money to be utilized for something else, for some consumable or something. That flexibility lies with the PI, which is not the case with other grants. So the grant is very strict on whatever budget they have allocated. But this is one grant which I think it's very tough to get it, but it's not impossible. Very they give this grant every year twice, and you crack it, at least you are fine for 5 years. And you have the flexibility to use the funds with the change in time. Like you are not bound to whatever you have written 5 years back.
Okay. So it's look like a program type. You can change the
Yeah,
things, but the
yeah,
direction is fixed in the
yeah.
same
Like the
direction.
objective is same, but the heads can be moved. Like if you don't have manpower, so that money you can utilize for reagent.
Consumables or
Yeah,
yes.
so something else.
Hmm. And definitely, if you got a fund, then you got the manpower. And sometimes labs are full of manpower. Very very interesting to see a lab with different type of peoples, different ranking, JRA, SRA, project assistants, and all. So now I wanted to know your views or your experience on people management. Means must be there is conflict because you also do the basic research, also the commercial part of invention. So it is also in your mind that how to manage the time, how to manage work-life balance, and if there is some conflict of your team and group. Then how it is affect you and how you managed these situations.
Yeah, as you said, when you have many people from many background and you have forget about many diverse background. If you have more people, it's easy to have conflicts, and it happens anywhere. I don't think it is new to any lab or any work environment. But the only thing, like there are situations where people have conflicts, but one thing I make sure, like the work should go on. Like whatever conflict, it should not be a personal conflict. Personal conflict should be out of the lab. But if you have a conflict in the lab, suppose you imagine like you are going to use the centrifuge and someone is already using and booked it. And you say, okay, I need the centrifuge now. And he said, no, my work is important. I need to use it first. So then you have a conflict, but it is a temporary thing. And that conflict is good, some in a way that people care for their work. If they don't care, they will not fight for doing the centrifuge. Right. So that means I need to get another centrifuge.
Mm.
Then the problem is solved, right? So, but in some cases, in my personal view, it is like put both the people together, or if 3 people are involved, 4 people involved in a conflict, let them say their sides. And I think that's the best way to solve it. And eventually you figure out no one is at fault. It
Yes.
just happened the way someone said something. It is never happened because someone did something wrong to the other person. It is always, I felt like it is just the way you are communicating with me or I'm communicating with you. That led to the conflict. If you say the things nicely, probably like the other person will not feel that bad and you will never have a conflict actually. And these are very common problems and they get solved quickly. And sometimes I don't even have to bother, like they solve it and people are mature enough uh, uh, to solve these issues. And the only thing I make sure is the mandate is same. We are working for science and we should work together. That is the thing I make sure they do. Whatever personal differences you have, that should be out of the uh, work environment.
Yeah. And yes, and they are doing the PhD and research, so definitely they
Yeah,
are mature. they mature.
And
And even,
you said management, like I really don't want to manage them. Like even for like suppose you say keep whether they are coming or not, or whatever, like every day what they are doing, like these are micromanage. I don't do that. Like these are mature people and they know what to do with their life and what to do with science. And I don't want to be a like boss sitting with a hunter. Okay, every day you have to come at 9 o'clock, do this. And for science, it is not helpful. And the way I have been trained is different. Like I don't want to manage people. I just want them to do the right thing they want to do and they believe in. And many times I also tell them like what project do you want to do? Like there is some kind of flexibility. Although we are bound for the grant, but there is also flexibility to do things what the student is interested in, so that they keep themselves motivated. I don't have to motivate them every time to do the job.
yeah, and that is the good situations. Means everybody should love their work and
Right.
do their own space and time, because otherwise it's science. It's not some mechanical work, or
Yeah, right.
it's not a civil work. It's a science, so it's mental peace, and it's also matters to do the work, do the right calculations,
Right.
right employment. So yes, I understand you not focus on the micromanagement. You will keep them tasks, and they are doing on their own. Sometimes you interfere when the situations is conflict. And what was your criteria to select a manpower, or maybe what is your criteria to collaborate with another scientist? Then what things you are looking for before the selecting someone or connecting to another scientist for collaboration?
Yeah, so basically, like, say, in the interview, like you call a person for 10 minutes, 15 minutes, or maybe half an hour. But it is not about, like, I don't judge them based on their credentials or the CV, what they have written, how many techniques they know, and what they have done. Because I come from same background, having no experience, so I never look at their CV, what expertise you have, what skills you have. But uh, only thing I look at is whether this person is a good person or not. The first thing, whether I can connect with this person. Can I work with this person in a long run? If you like you say like you always have a vibe, like when you talk to someone and then you realize, okay, this person is good. Then if you know, okay, this person is good and we can have good terms, then other things can be worked out. If he doesn't have paper, he doesn't have skills can be like taught. He can go for a training and learn things, or he can learn from the seniors, or whatever way. But he should have enthusiasm to join and to do the job. And he should be aware of what the things are going on. Like some people come for interview without knowing what is going on in the lab.
Hmm.
So that is not what I look for. I look for people who really did their homework and they're prepared what is coming next. And if they don't have skills, it's okay. But I look for people whom I can work with. And same with the collaboration also. Like for collaboration, you need to have good scientific combination. Like if we have complementary expertise, that is the first criteria for any kind of collaboration. Like if we are either me contributing to his or her project, or he or she is contributing to my project, which I don't have, or which he or she doesn't have. Like it should be complementary. when it comes to collaboration, because everyone cannot be expert in all fields. So we need support from others always, because even many times we don't know many very small things, and those things also are important for running a project or getting the things right. So those complementary things are very important for collaboration. And also uh, it is the person, at the end of the day, if you can deal with that person in a long run. Science comes later, like first we are humans, then scientists, right? So I care more for the human side, but and then the science things come later.
Yes, and as you said, because why I put these questions, collaborations nowadays very important, because if you any good paper in science, there is some paper which has 300 authors, 300
You,
plus
you,
authors.
you, you need that. Yeah,
Yes,
without collaboration, you cannot have
huh,
a good
yes,
paper.
and what I think or feel that in developing countries like ours and our neighborings, we are not so good in collaborative work. Where somehow we have some ethical or some predetermined mental some issues that we are not going to collaborate in that way. But yes, complementary experience or complementary setup of experiment will be one main strong make this bond so strong, because it's we needed that.
Yeah,
He needs us, or I need them. So the needs is men. What I think the
yeah,
need
right. The
is,
need, yeah, if you
yes.
need a good paper, you need
Ah,
collaboration, because you cannot do everything in your lab. You don't have
mm,
expertise.
yes,
And
yes. And
yeah, so,
sometimes what happened, maybe you have a expertise, but you don't have a setup right now.
or you don't
So,
have money to do it.
yes, yes.
Yeah,
That is
so,
that is sometimes happen that okay, I know these things, but right now
yeah,
I can't do this.
I can do this.
So it's also situations where you need a collaboration. So yes, collaborative work is nowadays very important. It's not only validate your work, but it's also validated by someone else to
Yes,
write or
correct,
not. So it's
correct.
also a validations is also
Hmm.
give some importance to
Right, right. Yeah. And
and
even now, government is forcing for grants more on collaborative basis, like not individual
yes, yes.
grants. Like you should have consortium kind of grants where like multiple people, multiple institutes are involved, so that like it should look like a mission kind of program. It is not just one person working on one problem to solve it. It should be a mission mode thing, to like many people working on the same thing and coming up with a better solution.
Yes. And now I want to ask you a question. Maybe many people are thought on that, that you did a private, you able to build a company. It's a private limited company and commercialized your research. But it is not possible for everyone, because I know it's lot of challenges and lot of officials work, lot of things you need to do before starting a company. So, can you help us to understand what are the other methods of commercializations? Means how we can commercialize our research other than from a private limited.
Yeah. So yeah. So this is one way I did. Like this is like you give your technology to a company for
Mhm.
commercialization. The other way is like you file a patent or file a trademark and give it to a completely different company. You are no more involved in that, and the company takes care of the development. of the product. And in this case, like if since I am involved in this company, so as you said, like there are many not challenges, these are like learning experience. As a scientist, I don't understand business, right? And how to have a, whether it is a private limited company or any other type of venture I need to do. So those things I learned, like what to do. But like you said, like the other way is just give it to any company for their development, their commercialization. You say, okay, we have this technology, we are selling this technology. Either you take a like one-time charge, the not you, basically the institute takes a one-time charge, or they say, okay, one time we are taking this much price. Once you develop the product, we'll take the royalty from the profit, whatever you are making, some percentage. So that way it goes. And yeah, like now government is promoting this entrepreneurship things, so that like more and more things are made in India uh, for the betterment. Yeah.
So, okay, and as you got many funding for, do you have any do you have some? Do you please give you us some idea on the available funding for entrepreneurship? If there is some specific funding in India West mainly.
Okay. Yeah. So I think if I can share my
Yeah,
slides,
sure, sure.
yeah, I think I have something to show there. Um, it will be very specific to like India, and mostly for the. Can you see my screen?
Yes.
Yeah. So it is. This slide is not specific to India. It can be for anyone. Actually, like when you start a startup, like you need funding, and the funding mostly usually comes from the founders. You have to put your own savings, because no one is going to believe your idea and give you fund except yourself. If you don't believe in your idea, but like why someone else should believe and give you money to start it. So the first fundings is usually the funders, and if the idea is brilliant, then some investors can come in to fund it. And the other options are like you get the funding from family, friends, and there are government grants and seed funds for the startup. And like mostly like after mm, this Modi government came into power, they really wanted to have Make in India program, so that we make everything, we don't import things. So they have Startup India, and also Odisha has Startup Odisha. So they have seed funds for and grants for the startups, and of course the investors, and even the banks. They also now realize that the startups are the best place to invest, because although many startups fail, but if the startups succeed eventually, the return is huge. So they are also willing to give loan, and even eventually, if everything goes well, you can go for public offering. And the first thing for a startup to start, like for the biotech sector, we have BIRAC. This is part of Department of Biotechnology, Government of India, and Pioneers. So they fund. They have a lot of funds for biotechnology startups, and many of the Indian, participants might have heard about uh, uh, PEAK, that is Biotechnology Ignition Grant. That is one of the best grant in biotechnology. Apart from that, there are other things from DST. There are grants from MSME. There are also awards and fellowships for students, and also like the startups, like Startup Odisha. Every state government also have come up with their agencies to promote the startups in by giving fundings or by giving money in the form of debentures. So, so there are many opportunities. If someone really wants to go into making a company, they can uh, find many opportunities. Like you should apply many agencies and see whoever likes the proposal, and I'm sure so one of them will be eager to fund if the idea is good and it is fitting to their mandate. As I said, BIRAC is for biotechnology, but there are other agencies like Startup India. They that is for any field. They have seed funds. They have funds for debentures. So funding is available. So it is not that people cannot get funding. If the idea is good, they can find a funding partner.
And sometimes getting the fund at the right time is a challenge. Some startup we try to getting fund for last 3-4 years. Maybe somehow it's not clicked because not it's
Right.
not for everyone.
Hmm.
So maybe it leads to stop their journey in very beginning, and not they are going to be for a long period without getting a fund, because as you already said that founder need to be invest their money, but there should be a limitation. No? Means
Yeah,
they should
correct,
be
correct.
limit
Oh,
their themselves from a certain stage.
yeah.
So what do you think on that? Status fails in early. What happened? Means if it is in that way, then how it's helping our country if they are failed in very initial stages?
Yeah, so see, like it is not only in our country. If you see US statistics also, like about 90% startups, uh, they fail. No, not exactly, I won't say they fail. They just didn't succeed. Uh, but as you said, like how it is helping the country. Like, see, you put your money to start.
Hmm.
But if the idea, it is not like you cannot put unlimited money to support the startup. But to start the business and to have a like a product or a prototype to showcase to the grant agencies that this is what I'm developing. And if the product is appealing to the masses, that is very important. Like someone should look at the market, if the market really needs the product or not. If the market needs the product, why a grant agency shouldn't say no? They can say no if there are similar products and there is no improvement. But since an innovator is developing something, want to have a startup, they should have a idea to improvise the existing product. And I think the grant agencies will uh, find it interesting, and they'll be happy to fund. And I missed the last part. I forgot what was
Uh,
that.
no, it's you answered, because the situations means most of the startup are
Okay,
failed.
yeah, correct. So
And
so
how?
most of them fail, you said.
Uh,
Yeah, but it's not failing.
hmm,
It is an experience. Like, see, if my startup fails tomorrow, I won't blame me or the idea or the funding agency or anyone. It is an experience. Like now I know how a company runs, how to make a company, and what are the like pros and cons. So in future, if I want to do something, I will not do the same mistakes again. Right. So it is and for the country, as you said, like although many of them failed within 5 years, but the products they make and it is suppose our startup India, like how we are helping the country. Like if the startups are making something and we are not importing, so there are two ways we can save the money for the country or increase the GDP. Either you make your own product, export it, get the money from outside, or you don't spend your money by buying products from abroad. So you make your own things and you don't spend the foreign reserve. So that way the startups are helping the economy to grow. Although some of them fail, like a big competitor comes into market and the startup cannot sustain, that is okay. That is part of the game. So we should not be worried about like we are going to fail, so we should not start. No, it is an experience. If it doesn't succeed, it is okay.
And if there is some similar product is already available in market, then why should someone start a startup when the similar product is already in market?
Yeah, I think like it is mostly like, as I said, if the similar product is there, whether it is made in India or it is coming from outside.
Okay.
For biology research, if you see, most of the things we buy from outside. Right? So although things are there in the market, but they're not made in India. And because of that, we are losing a lot of money from India. So if those things can be made in India, then we'll save a lot. And that will eventually increase our GDP. Even if we don't export anything to outside, at least we will not spend our country's money to buy things from abroad. So that is a saving for the country.
Yes. Yeah. So I think we covered every. I tried to bring the all related questions because it's also everyone's mind, and I'm also sometimes thought about this type of thing that for a scientist to start a business is very difficult, and there may be lots of challenges. So just we are at the end of our session, and we're going to end. And for the attendees, I'm just giving a link for feedback, and it also serves as a e-certificate applications. So those are registered, only get that certificate if they are fill this feedback form correctly. We match their email ID. If it is matched with the registration and feedback, after 2 days, certificates available on online. So I'm going to give that link in YouTube live chat, and it's only available on YouTube live chat. Please don't mail me or WhatsApp me or Telegram me for the link. It's only available on YouTube live chat. So at the end, just we want to conclude. We just wanted to know from you. I'm sure you're enjoying this challenging thing, because sometimes challenges motivate us. It's give us some some foods on our mind also, because we all scientists are love challenges. So it's you just enjoying it. But just last question to you, and in this webinar, that how you manage everything, because you have a family, you have a company, you have a lab, you have a many students. Those in early, well, means they are final stage of their PhD. So must be their publication, their thesis checking. So and also product promotion, a lot of UCSC from your fundamental research grants, from your product commercial research grants. So how you manage everything? Just thought on your management, or what is your philosophy on that?
Yes, so like everyone has this 24 hours, and it is all about the priority. And as a scientist, my priorities are my students. Basically, as you said, like uh, my students' thesis and this thing. Like last few months, we have graduated to PhDs. So their papers has to go, and the thesis need to be checked, and then thesis has to go for evaluation, and they have to be awarded. So that is the priority. And the product development, I said, that is a byproduct of the basic research. So that is also a kind of like a part-time thing for me. It is not something, and the company is a private limited company. It is so as you know, maybe some people may not know, like you have different type of companies. The one person company, you have LLPs, you have private limited companies. So private limited company is not a company where company cannot run without me. I'm part of the company. If tomorrow I exit from the company, company will still go on. It is based on the share people have in it. If say a big investor comes in, they will take over, and I will be out of the company. But the lab will stay, and that is the priority. So it is all about the priority. And product, yeah, it's a startup company. It is not a huge company. I need to spend time for that thing. It is a startup, so it is just few year old. So I don't really think it is a problem to manage two things. Like instead of 5 students, you can imagine you have 2 more. So so it's okay. So it is not a big deal to manage. Yeah, of course, you said the family thing. Yeah, that is sometimes like you have to compromise, because you cannot compromise on science. You cannot compromise your students' thesis. But one thing that is most of the time we compromise, I guess everyone does, with the time you cut from your family. That is the only thing how you manage, because they will understand you better. They know, okay, you are doing something, so it's okay. So, but for that, like, I would thank my family, like, for to support that, because without their support, I don't think it's possible, because you cannot cut time from any other place. That is the only place where it can be adjusted if they are agreeing. And thankfully, that is the case. And yeah, so we always say work life. So work comes first. And yeah, ideally it should be equal, but usually I don't
It's not
think
possible.
it really happens. And at this stage of the career, I don't think we really, not me, you, everyone. Career comes first and your work comes first. But somehow you have to prioritize and manage. And yeah.
Yes. And definitely, families give us the mental peace so we can do better work in our professional life. So family contribution is the most important for each and every researcher lives. And thanks for today's webinar. And it's it 870 people are watching right now. So
That's
uh,
great.
we I just include one thing. Uh, Amrit sir always looking for the students, either for the his research or his company. So those are interested to join a job or lab, you should communicate with Dr. Panda. I'll put all the
Currently, we have openings, and it is already advertised in ILS website. And
okay.
we have also openings in the company also.
So yes, I know. You are the one people who are always looking for the students, and manpower. So I request people and those are listening, or maybe a friend of if you're looking for a job, you may approach to Dr. Amras Panda's, and I will put his details on the our website, bioengine.com, after the webinar, and certificates will downloadable after 2 days. I'll put all the certificate in 3rd of July from the downloads. So, sir, it's a great talk. It's a great interaction. I'm sure lots of people are interested and they are comment, but I'm not taking all the comments because we are covered up. We are covered up everything. And based on their comment, I also formulate the questions and all. So the end of our sessions is a great privilege for us, and we really enjoyed your thoughts, your research, and your journey, mainly your journey. We wish more success to from your lab and from your company, and maybe more products which benefit all of us. And thanks to accept our invitation and participate in Bioengine webinar series.
Thank you very much.
Thank you. Thank you, Bioengine, for this opportunity. And thanks to all the viewers. And thank you.
Thank you. Thanks. So it's end of our sessions. So please submit your form within 5 hours from now. I already shared the link, and I am going to once again share the link. And after 5 hours, there should be no communications about the feedback and all. And we try to bring another webinar end of this month, and it will be uploaded on our websites. So bye everyone. Have a nice day. Have a nice night. Bye.
Thank you, everyone.
Yes.
See you again in our next webinar. Okay, I will end the session now.
Okay. Bye.
Bye.